Hi, everyone. This is Christy Sullivan. I'm the secretary of the American Society for Cellular and Computational Toxicology, and I'm very excited to welcome you to this webinar series. This is our April webinar presented both by the European Society for Toxicology in Vitro and ASCCT. I would encourage you to visit our websites, which you can see on the screen, to learn a little bit more about both of our organizations and how you can get involved, and to consider becoming a member to help support activities like this. This webinar is going to be recorded, and the recording will be posted on the ASCCT website a few days from now. You can see an archive of all of the past webinars there as well. In the webinar platform, you can ask questions of the presenters using the Q&A feature at the bottom toolbar on the bottom of your screen. So please do put your questions in the Q&A, not in the chat. That will really help us keep track of all your questions coming in. And we will have a Q&A after each speaker. We have 2 speakers today, and so make sure you get your questions in while the speakers are talking. We are also going to try something new today where we have enabled automatic subtitles because some people find that helpful in order to follow along. If you want to enable them or hide them, you can do so on this toolbar at the bottom of your screen where it says live transcript. If you need any other technical help, please feel free to message us in the chat. And I would also encourage you to say hello, let us know where you're joining from, and join the conversation. So I just want to briefly mention a couple of upcoming events. First of all, ESTIV's annual meeting is going to be in November of this year in person in Barcelona. So check out their website. There is abstract submission is still open until a couple weeks from now. May 1st is the deadline. So please feel free to check that out, and hope you will participate. And we have also set the dates for the ASCCT annual meeting. That's going to be October 19th through the 21st in Research Triangle Park. It's primarily an in-person meeting with a virtual option potentially available. It depends a little bit upon the venue and what is available there. But abstract submission will open soon, so I encourage you to sign up on the ASCCT website for email updates, and then you will be notified of updates as the meeting is planned. And we also have scheduled our next webinar. It's going to be May 16th, and we're going to have 2 nice presentations on genome toxicity and cancer tissue culture. You can go to the webinars page to register for that webinar. And then I will go ahead and introduce our speakers for today. So the first speaker is going to be Sabina Burla. She is the Research and Development Engineer at the Luxembourg Institute of Science and Technology, and a PhD candidate in biotechnology at the University of Agricultural Sciences and Veterinary Medicine in Romania. After earning her MSc in PharmD, she worked as a research assistant for the Platox project. Her current work centers on assessing the effects of exposure to pollutants and the development of relevant NAMs for respiratory sensitization. Sabina's work has won the ESTIV Best Poster Award from the 11th World Congress on Alternatives and Animal Use in the Life Sciences, which is awarded to a first author, PhD student, or early career scientist by ESTIV, presenting their work at the 11th World Congress last August. Sabina also most recently won the In Vitro and Alternative Methods Specialty Section Graduate Student Award at the SOT annual meeting. And our second presenter is Dr. Kelly Carstens. Kelly earned her PhD in neurobiology from the University of North Carolina, Chapel Hill, where she focused on studying the molecular mechanisms underlying adverse outcomes in mice modeling neurodevelopmental disorders. Her interest in reducing the use of animals in toxicity screening led her to join the US EPA as a postdoc and further study the application of computational approaches to evaluations of developmental neurotoxicity. She is currently a principal investigator at EPA in the Center for Computational Toxicology and Exposure. And Kelly won the Ed Carney Predictive Toxicology Award, which recognizes the first author of an outstanding poster or platform presentation that will advance predictive toxicology, and is named after Dr. Ed Carney. Some of you might remember, who was a member and supporter of ASCCT in its early days and a leader in predictive toxicology. Kelly was given this award for her presentation at last October's annual meeting. So I will now give the floor to Sabina Burla. Sabina, please go ahead.
Hello, everybody. I hope you can hear and see me, and you can see the first slide of my presentation.
Everything looks good.
Thank you so much. Good morning, good afternoon, and good evening, as we have a very wide audience from the whole planet, I see in the chat box. Thank you, Christy, for the nice introduction. I will just introduce myself. I'm working at Luxembourg Institute of Science and Technology. I'm mainly working in respiratory toxicology with a focus on non-animal methodologies for respiratory sensitization. Throughout the presentation, I will briefly introduce you in the topic of respiratory sensitization and the status of existing methods for the purpose of evaluation and classification of chemical respiratory sensitizers. I will present you the objective and the workflow of the study, the materials and methods that have been used, the results that were obtained, some conclusions, and some future perspectives. So let's go and dive into the presentation. The chemical-induced respiratory sensitization may lead to the development of allergic pathologies in the respiratory tract, among which I am mentioning the allergic rhinitis, the rhinoconjunctivitis, and asthma, which have become a global public health problem in the last decades. As opposite to skin sensitization, for which the immunological mechanism has been completely unraveled and an adverse outcome pathway, an AOP, has been fully described, the immunological mechanisms for respiratory sensitization caused by chemicals are still under investigation. Moreover, despite the fact that skin and respiratory sensitization present mechanistic similarities, there are differences which lead to distinct adverse outcomes in the two distinct adverse outcome pathways on the two distinct AOPs. Therefore, the existing validated in vivo and in vitro test system for the identification of skin sensitizers fail to correctly classify respiratory sensitizers because these methods cannot differentiate skin from respiratory sensitizers. There are currently no validated or widely accepted test methods for the prospective identification of chemicals which might induce respiratory sensitization. The single advantage in this unfortunate situation is the possibility to develop non-animal methodologies, NAMs, for the identification of respiratory sensitizers, thus reaching the goal of 1R, the full replacement of animals in science. Therefore, at least a three-dimensional alveolar in vitro model for the prediction of respiratory sensitization was designed by Dr. Alim Shari and the team. The detailed build-up, the full characterization, and the performance of the model to discriminate sensitizers from non-sensitizers is available in the paper indicated on the slide. In this study, 2 respiratory sensitizers and 2 irritants were evaluated, and the main objective of the current study I am presenting now was to test the performance of the model to differentiate the respiratory sensitizers from irritants using an extended panel of test items, including 10 chemical compounds. The test system offers several advantages. The first one that I can mention is the fact that it can be exposed under realistic physiological conditions at the air-liquid interface at ALI, by contrast to the classical submerged exposure, which does not mimic the physiological exposure of the lungs. Moreover, the test system allows the development of a tissue-like microenvironment similar to the alveolar barrier, which is shown in Figure 2, directly by cell-to-cell communication in the system, but also indirectly through the secreted messenger molecules. One of the most important features of the test system is the activation of the antigen-presenting cells, the dendritic-like THP1 cells, following the exposure to respiratory sensitizers, which was proven by the increased expression of cell membrane markers on the surface of dendritic-like THP1 cells. The clusters of differentiation CD54 and CD86 on one hand, on the other, the receptor for thymic stromal lymphopoietin cytokine, TSLPR. This is an overview of the complete workflow of the study, and I will start to develop it. To test the capacity of the test system, 10 chemical compounds were chosen, among which the first 6 are respiratory sensitizers belonging to different chemical classes. We have chloramine T, ethylenediamine, piperazine, glutaraldehyde, ammonium hexachloroplatinate, and maleic anhydride. The next 4 compounds are formaldehyde, glyoxal, salicylic acid, sodium dodecyl sulfate, and these are the irritants. The choice was based on the available information from the scientific literature on the topic of respiratory sensitization and the data provided by the European Chemicals Agency in the chemical substances registration dossiers. The panel of test items is presented with the name, the chemical structure, the molecular weight, which is a very important feature for respiratory sensitizers as they are mainly low molecular weight compounds, and also the vehicle in which the stock solution was prepared. The test system is built using human cell lines. We have A549 cells, which represent the type 2 human alveolar epithelial cells with the ability to produce surfactant, a very important feature in the test system. The EAHY926 cell line, it's a representative for endothelial cells, which form a single cell layer that lines the blood vessels and regulates the exchanges between the bloodstream and the surrounding tissues. And last but not least, we have the THP1 cell line, which is a human monocytic leukemic cell line. Table 3 gathers all the information concerning the complete cell culture media used for the maintenance of the cell lines, but also for the buildup of the test system. The experiments are started on day 0 with the differentiation of THP1 cells to macrophage-like THP1 cells using 4-bol-12 myristate 12 acetate, abbreviated PMA. On day 2, the actual buildup of the test system is started by first seeding the endothelial cells on the basolateral side of the hanging cell culture insert, and 4 hours later, the A549 cell suspension is seeded on the apical compartment of the insert. On the same day, the PMA from the macrophage-like cells, which attach to the cell culture flask, is removed by aspiration, and fresh THP1 complete cell culture medium is added. On the 6th day, the dendritic-like THP1 cells are seeded in the wells of a 6-well plate insert. The inserts are moved to the plate, and the macrophage-like cells are detached from the cell culture flasks and seated on the apical surface of the insert. Ali is created 4 hours later by the complete removal of the cell culture medium from the apical compartment of the insert by gentle aspiration. Inserts are left for 24 hours in the incubator at air-liquid interface, and on the 7th day, they are exposed at the air-liquid interface using the Vitrocell Cloud 6 system. This system is fitted with an aerogen nebulizer, as shown in figure 5, which is provided with a vibrating mesh membrane that generates aerosols from liquid solutions and suspensions in a closed chamber, the exposure top, as shown in the video. The cloud of aerosol settles down within a period of 15 to 45 minutes, depending on the vehicle control that it's used for the preparation of the stock solution of the test items. The plate with insert is placed on the base plate, which is connected to a module heated up to 37 degrees Celsius. The first step in the study was to perform a dose finding assay to calculate the CV75 value, the concentration showing 75% cell survival, in order to determine the optimal concentration of the test items for the CD54, 86, and TSLPR cell surface markers expression measurement. Research work that led to the development of the human cell line activation test, the H-CLAT assay, has shown that some skin allergens must induce a certain level of cytotoxicity to increase the expression of cell surface markers in the dendritic cells. Following the 24-hour exposure to the test items in a dose-response curve manner and corresponding negative controls, the vehicle controls, the cell viability of the test system was evaluated using the Resazurin assay, which determines the metabolic activity of the cells as a quantitative measurement of cell viability. The plates with inserts supplemented with resazurin working solution were incubated at 37 degrees Celsius, 5% CO2 in the dark for 1 hour. After the incubation time, the aliquots of resorufin were sampled from the apical and from the basolateral compartments, and the fluorescence of resorufin was measured using a multi-mode microplate reader. In addition to the resazurin assay, the cytotoxicity of dendritic cells alone was evaluated. The THP1 cells were stained with cytox blue, a high-affinity nucleic acid stain that easily penetrates the cells with compromised membranes. 24 hours post-exposure to the exposure dose leading to 75 cell viability of the test items, the THP1 cells were collected, stained with fluorescence-labeled monoclonal antibodies against CD54, CD86, and TSLPR cell surface markers, and the isotype matched controls, and they were analyzed. The cell viability and the expression of the cell surface markers was analyzed on a Fax Celesta flow cytometer using the BDFAX Diva software for data acquisition and the FlowJo software for data analysis. On this slide, I am presenting the dose response curves on the cell viability corresponding to the apical, to the basolateral, and the complete test system. The experiments were performed in at least 3 biological replicates and in 3 sprays. The data is expressed as mean, and the bars represent the standard errors. Here, in the same manner, I am presenting the dose response curves for the irritants. The CV75 value was finally calculated using the OECD test guideline number 442E formula for the apical basolateral compartments separately, and also for the complete test system. Based on the cell viability, the cytotoxicity data, and the calculated CV75 doses, the final exposure doses were defined for the cell surface marker endpoint measurements, and they can be seen in table 4. So the expression of the first evaluated cell surface marker was for CD54, and this one was significantly increased following exposure to ammonium hexachloroplatinate. The relative mean fluorescence intensity of 381% is above the threshold of 200%, marked by a red dotted line, threshold set for the H-clat assay at which a compound can be considered a skin sensitizer. All the other respiratory sensitizers induced an upregulation of the CD54 cell surface marker under the 200% threshold, chloramine T to 115, ethylendiamine to 117, piperazine to 136, glutaraldehyde to 182, and maleic anhydride to 159%. The respiratory irritants also induced an upregulation of the CD54 cell surface marker under the 200% threshold, with the exception of sodium dodecyl sulfate, which induced a downregulation to 86% by comparison to the vehicle control. The expression of the second evaluated cell surface marker on the THP1 cells, CD86, was significantly increased following exposure to the respiratory sensitizers piperazine, glutaraldehyde, and ammonium hexachloroplatinate. No significant increase of the relative mean fluorescence intensity was observed following exposures to the respiratory sensitizers chloramine T, ethylendiamine, and maleic anhydride for the CD86 cell surface marker, but ethylendiamine exposure led to a slight increase of up to 165%, a value which is above the threshold of 150%, marked by the red dotted line, threshold set for the H-clat assay at which a compound can be considered a skin sensitizer. Chloramine T and maleic anhydride were the respiratory sensitizers that did not increase the expression of CD86 cell surface marker above the 150% threshold. All irritants showed values below the threshold of 150%, and sodium dodecyl sulfate induced a downregulation to 89% by comparison to the water vehicle control. The third investigated cell surface marker, the receptor for TSLP, has been shown to be involved in the respiratory sensitization and could be a suitable marker for the prediction of respiratory sensitization potential of chemicals. In this study, we set tentative criteria for the identification of the relative mean fluorescence intensity values of the TSLPR cell surface marker. The tentative criteria chosen are the ones proposed by Sakaguchi and the collaborators in the development of the H-clat assay. 120% shown in the green line, 150% shown by the purple line, and 200% shown by the pink line. In our study, only chloramine T, ethylenediamine, piperazine, and maleic anhydride exposures have induced statistically significant upregulation of the TSLPR cell surface marker, with values of 234 for chloramine T, 325 for ethylenediamine, 344 for piperazine, and 351% for maleic anhydride. For TSLPR expression, the relative mean fluorescence intensity values for all respiratory sensitizers were over the tentative threshold value of 200%, except for glutaraldehyde, which upregulated the expression up to 193%, and ammonium hexachloroplatinate with a value of 168%. They showed values under the 200% tentative threshold as mentioned, but still above the 150%. For the respiratory non-sensitizers, all values were under 150%, except for formaldehyde, which induced a slight upregulation of 161%. Salicylic acid and sodium dodecyl sulfate induced a downregulation in expression, showing values of 97% and 87% respectively. Formaldehyde is one of the irritants. It induced a slight increase over the 150% threshold. Despite the fact that formaldehyde is considered an irritant, in our study, it can induce a mixed Th1, Th2 immune response, being at the same time a skin and a respiratory sensitizers. Moreover, based on the chemical structure, aldehydes are expected to be respiratory sensitizers. According to the cell surface marker data, we can conclude that CD54 and CD86 cell surface markers alone do not allow the discrimination between respiratory and respiratory sensitizers and irritants, but the TSLPR marker has the potential to correctly identify all the respiratory sensitizers that were involved in this study on the 150% tentative criteria for TSLPR marker upregulation. To sum up, the data show that the in vitro alveolar model can discriminate respiratory sensitizers from irritants based on the increased expression of the three cell surface markers, TSLPR, CD54, and CD86. The model correctly identifies the proheptans, ethylenediamine, and piperazine as respiratory sensitizers. These are chemicals which have been reported as false negatives in other NAMs. We are assuming the possibility of the system to correctly identify the proheptans because of the metabolic activity exerted by the epithelial cells, the A549, in the test system. The cell viability data and the increased expression of the evaluated cell surface markers on the dendritic cells demonstrate the capacity of the in vitro model to mimic the complexity of the alveolar barrier. This is what we have done so far, but in the future, additional respiratory sensitizers, irritants, and inert chemicals will be tested to evaluate the false negative, the false positive results, and as well the skin sensitizers need to be tested to evaluate the potential of our test system to discriminate between the respiratory and the skin sensitizers. At the same time, it is necessary to evaluate further endpoints like cytokine, chemokine secretion, and gene expression in specific cells in our test system. Thank you very much for your attention, and I would be happy to answer your questions.
Thank you, Sabina. That was a really lovely presentation. You covered a lot of material in a few short minutes, so thank you. We have quite a few questions, and I'm going to try to cover a few of them. One of them is, could you explain the CD54 and CD86 measurements mean for the system, and what's their importance in the context of irritant versus sensitizer exposure?
Thank you for the question. So for the CD54 and CD86, this has been of course, first done in the skin sensitization assays, and it has been proven that it can be a meaningful marker for respiratory sensitization. And this is the question more related to technicalities or?
I think they're just wanting to know, are these are immune mediated?
Yes, so yeah, so practically we are seeding the dendritic cells in the basolateral compartment of the test system in the well of the plate. We are collecting the cells, we are processing them, and then we are labeling them with monoclonal antibodies. Of course, we are also using the isotype controls to make sure the experiments are going well. And then we are doing the flow cytometry measurements and then the analysis using a specific software, in our case, the FlowJo. And literature shows that these markers can be used also for respiratory sensitization, but from my perspective, they should be used all together with other endpoints, because then we cannot really make the differentiation between respiratory and skin sensitizers if we use only these markers. And if we are looking at the gold standard, the skin sensitization assays and the AOP, of course, there are a lot of assays that are being used to really put a chemical into the category of skin sensitizers, sorry.
A question about your model: is the surfactant added or is it produced by the type 2 cells?
The surfactant is produced by the A549 cells, and this can be seen in the publication by Aline Shari. It was showed on the slide, but if interested people will search for her name, Shari, they will find the publication, and I think in the supplementary material of the paper, it's shown how it's evaluated, the production of the surfactant.
I think we can also add it to the webinar page. We've
Yes.
done that in the past. We can just put the publication up there so people can check it out.
Sure, it's an excellent reference.
A couple of questions about the chemical exposure of the cells, or actually a statement and a question. So I'll see if I can get your comments, because I know you have a lot of experience now with that. And so one of the comments is, the air-liquid interface provides a realistic exposure. However, for reactive chemicals that might react with water, it's important the liquid phase is like lung surfactant and not just aqueous medium. And the commenter is suggesting the addition of albumin and glutathione. I don't know if you have a reaction to that. But there's also a question about formaldehyde being a gas, and they're wondering, have you tested it as a gas, or do you dissolve it into solution?
We solve it. So we are using a solution, and we are preparing dilutions in order to expose through nebulization. So practically, we are not really we are in a way evaluating the gas, but we are not starting from a gaseous phase. And indeed, for the statement, I totally agree. There are chemicals with a very short lifetime, which are hydrolyzing in water, or they are polymerizing, and these are considered difficult to test chemicals. Of course, it's a lot of work ongoing, and I agree that there are difficulties. This is just the nice part of the work. The difficulties are will be available somewhere in the future, and how we tackle them.
There are a couple of questions that I'm going to let you answer in text, because I think they can probably be answered pretty easily. But maybe one last question about the thresholds that you chose. I know that you explained for TSLPR that you got that from the HCLAT developers' work. But the previous ones for, I think there was 200 and then 150 for the 2 CD markers. How did you choose those thresholds?
I just chose the thresholds that are presented in the test guideline. It's okay. Nothing that I chose by myself. It was already chosen.
Okay, great. All right, I'm sorry, we have some other really good questions, and I would encourage you all to check out the Q&A to see if Sabina can answer some of them by text, because we're going to need to move on to the next presenter now. Thanks a lot for your questions, and thanks, Sabina, for your presentation and responses. And
thank you for the invitation.
Kelly Carstens, I will provide the floor to you.
Hello, are you seeing my slides?
Yes.
Fantastic. Thank you for the opportunity to present my work, and thank you, Christy, for the lovely introduction. So today I'm going to tell you about evaluating in vitro new approach methods for developmental neurotoxicity. So conflict of interest statement, the views expressed in this presentation are those of the authors and do not necessarily reflect the views or policies of the US EPA. So I'm going to start off by giving a little background on developmental neurotoxicity new approach methods. So neurodevelopmental disability is one of the most prevalent chronic medical conditions encountered in pediatrics, affecting about 17% of 3 to 17-year-old children in the US. Both genetic and environmental risk factors have been identified as underlying causes driving this prevalence. And environmental risk factors can refer to not only chemical exposure, but also early life infection, as well as maternal stress. So this has really motivated a lot of research in the area of DNT, considering both the environmental and genetic determinants that may be associated. Outlined by the US EPA guidelines for neurotoxicity risk assessment, DNT refers to any adverse outcome of exposure to a toxic substance on the normal development of the nervous system structure and or function. So in an effort to improve toxicological screening approaches and address some of the limitations associated with in vivo studies, such as cost and animal use, the agency has implemented DNT NAMs. So DNT NAMs refer to multidimensional DNT screening assays that cover complex neurobiological space in terms of temporal, different time points in development, different key events in neurodevelopment, as well as cell types and different species. However, we face many challenges at the moment in terms of evaluating DNT NAMs. A few important ones are listed here. Limitations are such that no single in vitro screening assay can recapitulate all critical cellular events of neurodevelopment. Some compounds may disrupt specific cellular events at different stages of neurodevelopment, such as a chemical may affect proliferation that may not affect synaptogenesis downstream. And some neural cell types may be differentially sensitive to perturbation. So in contrast to evaluating DNT based on an individual in vitro screening assay, the work I'm going to show you today demonstrates the power of using an integrated analysis approach of a broad screening battery of DNT NAMs using computational approaches. So today I'm going to cover 3 questions. 1, how does a broad screening battery collectively inform DNT relevant bioactivity? 2, can we build a model to classify compounds that demonstrate in vivo DNT bioactivity? And 3, can we identify biological gaps in the current EPA battery and or Tox21 database? So which neurodevelopmental processes are we capturing in the current EPA DNT NAM battery that I'm going to be talking about today? So in this schematic, 5 main key events in neurodevelopmental are highlighted. I've added a red asterisk next to the neurodevelopmental processes that are represented in the DNT battery. Proliferation, apoptosis, neurite outgrowth, synaptogenesis, and network formation and function. And something that's really useful about looking at neurodevelopment in this context is that you can also appreciate that there are a number of neurodevelopmental processes that are currently not represented in the DNT NAM battery, in addition to different cell types, such as non-neuronal cell types, such as microglia, astrocytes, or oligodendrocytes. I will highlight, though, in this table, again, the asterisks represent the assays that are represented by the EPA battery. There are additional DNT assays that have been developed and have been used for screening chemicals that are not currently included, such as neuronal differentiation and oligodendrocyte differentiation, which we hope to include in this integrated analysis down the line. And I also like to say there has been recent emphasis on including additional assays such as microglia and astrocytes. So that's something we are particularly interested in the near future. So the two assay technologies that are included in the DNT battery that I'm going to talk about today are the microelectrode array technology, network formation assay, and the high content imaging technology. So the network formation assay records neuronal activity at multiple developmental time points in vitro, starting at DIV5 and collected at also 7, 9, and DIV12 from primary rat cortical neurons that were harvested at day 0 and were grown in a 48-well culture plate that includes 16 electrodes per well. Using this platform, we can capture 3 different general activity types, such as mean firing rate, which captures the number of action potentials over time, or bursting activity, which captures the frequency of rapidly occurring action potentials, or network connectivity, which captures the synchrony of the neural network between electrodes. And this really reflects a healthy development of synapses, which are the functional connections between these neurons. And the high content imaging technology is an automated data acquisition analysis system for chemical screening that measures a large variety of morphological features of different neurons. And so in this case, we're measuring neurite outgrowth, synaptogenesis, and neurite maturation, proliferation, and apoptosis as different morphological features in a 96-well culture plate from different cell types listed here, which include both rat cortical neurons as well as human cell lines. And I will say that each of these assays include their own cytotoxicity endpoint measurement. So in the data set, we have a total of 92 chemicals that were tested across all these assays listed here. So once all the data is collected, it was then pre-processed and pipelined through the ToxCast pipeline framework, where the data are normalized and curve fit to identify active or inactive chemicals. So and on the level 4 of the pipeline, the dose response is fit to one of 3 models, either constant, Hill, or gain loss model. And then on level 5, the winning model is selected as a binary hit call of 1 or 0 based on the fitting of the Hill or gain loss and the model curve exceeding an efficacy cutoff. So in this example here, the efficacy cutoff is 3 times the baseline median absolute deviation. And then we can extract a point of departure estimate. And in this talk, I'll be using the activity concentration at 50% of maximal activity. And this is an example of what one of our concentration response curves looks like from our endpoint measuring the number of bursting electrodes, where we can extract our point of departure as indicated here. So what does this data look like when it all put together? And how does it inform DNT relevant bioactivity? In this heat map here, you're looking at chemicals across the rows and the endpoints across the columns. And the color indication here is the darker green-blue color. It indicates a more potent bioactivity, whereas yellow indicates inactivity. And what we found across this chemical set of 92 chemicals that were hierarchically clustered is that there's a general on-off effect, and also a lot of clustering by assay technology, where a lot of the network formation activity endpoints fell into this cluster, and a lot of the high content imaging endpoints fell into this cluster. And we also see a lack of activity in our network formation endpoints that are measuring increased activity, which is something that needs to be further evaluated in the future. So given the strong clustering by assay and really the lack of general differential patterns of activity, we next evaluated the possible role of cytotoxicity in explaining this on-off effect pattern. So how do we measure activity below cytotoxicity? We refer to this as selective activity. And one approach is using an area under the curve metric, where instead of capturing the area under the curve, which captures both the potency and efficacy as depicted here up to the highest concentration tested, we instilled a threshold of the cytotoxicity AC50 as the highest concentration, and then capture the area under the curve below that. So now looking at the same data set, but looking at selective activity versus non-selective activity, we can see that we now see much more informative differential patterns of activity. And before I jump into looking at each cluster and how it might be informative, I also wanted to show that I applied an unsupervised machine learning approach called k-means clustering to really optimize the number of clusters that we're pulling out of this analysis, in addition to the hierarchical clustering. So this analysis demonstrated that there appear to be 5 main centroids. Therefore, we should optimally partition the chemicals along the rows in 5 different groupings, with an anomaly noted here of colchicine, which had a very high area under the curve across many endpoints. Okay, so what can we learn from this computational clustering approach? So this is our highest selective activity cluster, and it captured compounds that were highly selective for synaptogenesis and neurite maturation, as well as neurite outgrowth in the HN2 human cell line. And then moderate selective activity for proliferation, neurite outgrowth, and general network connectivity and neuronal activity. And it captured compounds that contain 4 metals, 3 organochlorines, and 3 neuroactive drugs, which really indicates that we're capturing chemicals that we expect to be active in the DNT NAM battery in this highly selective cluster. Something that's interesting to note here is that we have 2 assays that measured neurite outgrowth in human cells in the yellow indication here, as well as neurite outgrowth in rat cortical cells in blue. And this was informative for indicating that there seem to be some cell types or species-specific differences in this particular neurodevelopmental process. As you can see, it's much more selective in human than rat. And our second cluster, we captured compounds that appear to be highly selective for network connectivity and general neuronal activity here. And this group of compounds contain several neurological drugs that have several drugs with that are neurological targets, in addition to several organophosphates and metals. And an informative observation from this cluster is that we actually captured 2 compounds that demonstrate a very similar mode bioactivity profile, but do not share the same mode of action. So these 2 compounds are haloperidol, which is an antipsychotic that targets the dopamine D2 receptors, and deltamethrin, which is a pyrethroid insecticide that targets voltage-gated sodium channels. So what this is suggesting is that there are likely off-target effects occurring in these 2 compounds that might be shared. So the key findings from the selectivity analysis is that selective data is more informative for understanding biology and compared to non-selective data. A subset of compounds demonstrate cell type specific effects, such as the example of selective activity in neurite outgrowth assay using human cell line versus a rat cortical cell line. And that selective activity clusters do not necessarily appear to be explained by shared mode of action and may also account for off-target effects. So secondly, we asked the question, can we build a model to classify compounds that demonstrate in vivo DNT bioactivity? And so here in our analysis, we captured 53 compounds with evidence of in vivo DNT activity. And these are the citations for the 53 compounds. And for more information, you can see my recent publication. And 13 putative negative compounds. So generating a confusion matrix where we identify where do these positives fall in our selectivity analysis, such that are we capturing our putative in vivo DNT positive compounds in our selective activity clusters, and are we capturing our negatives in our inactive cluster down here? And based on this confusion matrix, we captured positives with a 74% sensitivity and a 92% specificity, where we captured 12 out of the 13 negatives in our inactive cluster. So interestingly, if you compare the sensitivity to running this analysis again, but instead using non-selective data, so data that includes cytotoxicity information, we have a much higher sensitivity of 93%, which is indicating that we're capturing more of our in vivo positives in our active bioactivity clusters. And then for the last part of my talk, I covered the question as to whether we can identify biological gaps in the current EPA DNT NAM battery, which is really getting at the questions, how can we better understand some of the false negatives that we were capturing, or the compounds that were in the inactive selective activity cluster that we expected to be positive in the DNT NAMs. So this is an example of kind of trying to get at the question of whether we might be capturing the expected target mechanism in the DNT NAM battery. So caffeine was one of our false negative compounds that we expected to be active, at least at high concentrations. And we considered the ideas whether we're actually capturing the caffeine receptor, adenosine A2A receptor, which is shown here from the Allen Brain Atlas to be expressed throughout the brain, and it appears to be developmentally dependent. So looking at this heat map here, you can see that we wouldn't expect the receptor to be highly expressed in the brain at embryonic ages, but that it increases in development postnatally. And you can also track this here with these images to see not only timing of the receptor expression, but also where it might be localized at higher expression. And in our assay, we're capturing the rat cortical region, so that would be this region here. And so this data might indicate to us that we may have not captured a region of the brain at the time when this receptor would be highly expressed. So this really drives the need for generating a baseline transcriptomic profile of these cell models, which is something that we are currently working on to better understand our false negatives. I also use an in vitro to in vivo extrapolation approach using high throughput toxicokinetic modeling to answer the question, are we testing at high enough concentrations in vitro? So to do this, we use the HTTK-R package to perform forward dosimetry, where we can transform external doses identified in the in vitro assay to internal doses modeled as a concentration level in human to evaluate their relationship. And then I also use allometric scaling to transform in vivo rodent doses from the literature to human. And what this data is showing here is that in this list of our false negatives with available information to run the IV-IVE analysis, that in 2 cases in manib and caffeine, their in vivo doses, which is indicated in the red and the purple triangles, appear to be either above or right around in line with the highest concentration we tested in vitro, which is the blue bullseye. And so this is just another way to get at validating why we might have seen a false negative, and might it be important to retest at higher concentrations in the case of certain chemicals that we would expect to see activity in the NAMs. And then finally, can we capture gaps in the current ToxCast-Tox21 database? So the ToxCast database includes over 1,500 assay endpoints that cover heterogeneous assay types and tissue sources, gene targets, and biological responses. I've listed several of the biological responses. This is not all inclusive that are captured by ToxCast to get an idea of the breadth of responses that are represented, and now including the DNT NAM data that I've talked about today in ToxCast will add a representative of neurodevelopmental processes in these biological responses. So what this data is showing you here is that if we look at the range of activity in ToxCast, which is indicated by these box plots here, and compare this bioactivity to the activity that we got from our DNT NAM assays, which is represented in purple. And this is specifically the 5th percentile of the selective DNT AC50s. So this is really capturing the minimal effects of the DNT NAM activity. What we can see is that in several cases, particularly methylmercury, mannab, and heptachlor, our DNT NAM assays captured activity that was significantly lower, so left shifted compared to ToxCast. So it's really demonstrating that the DNT NAM battery captures potent and sensitive effects relative to the general ToxCast activity, suggesting that neurodevelopmental processes are important to include in the biology covered by ToxCast for identifying health protective potencies. So in conclusion, I showed you that selective data is more informative in identifying differential patterns of functional activity than non-selective data, and that selective activity clustering do not necessarily appear to explain mode of action, to be explained by mode of action. And that to answer the question about whether we can build a model to classify compounds, the selectivity metric appears to be informative for classifying with high specificity and moderate sensitivity, and we get a higher sensitivity from non-selective activity. And we found that the false negatives from this analysis provide insight into experimental and biological limitations, which may be associated with factors such as cell type, species, or developmental time point represented. And that the DNT NAMs overall provide added value to the ToxCast Tox21 database from the perspective of capturing health protective potencies. And so with that, I'd like to acknowledge all the people that were critical in helping me develop this work, specifically Tim Schaefer and Katie Paul Freeman at EPA. And I'd like to thank everyone for attending this talk, and I'm happy to take any questions. Thank you.
Great, thank you very much, Kelly. That was really nice. I really like the focus on sort of evaluating where are the gaps and where can we, what needs to be filled. So we have a few questions to start off with. Sort of a specific question about the rat cortical neurons. Did you measure cytotoxicity at DIV 5, 7, and 9 separately?
That's a really good question. The cytotoxicity endpoint was measured at the end of the assay, which was at DIV 14. So that's something that's really interesting and important to consider, and it would be very useful to have data on cytotoxicity at each developmental time point. So that could be something to consider in the future.
Okay, great. How do you determine the validity of the DNT reference chemicals, and specifically related to caffeine? Is there any human clinical data associated with developmental neurotoxicity for caffeine?
Yeah, so I added this reference here to the slide. I strongly encourage anyone who's interested in understanding the list of compounds with evidence of in vivo DNT to refer to this publication. So there are several examples. I think the two in here are cited from papers in rodent studies back in the 80s with evidence of DNT associated with caffeine exposure at very high concentrations. There have also been more recent studies that I've read that have found changes in after gestational exposure of caffeine in BDNF and development of GABAergic networks. So so essentially, I think this is something really important to think about, and I would like to emphasize a lot of the limitations and assumptions that are made with creating a reference DNT positive list. A lot of the in vivo adverse outcomes that are captured in this list are all over the map, I guess, in terms of there might be representative adverse outcomes of structural changes. There might be behavioral changes in rodents. There might be human epidemiology studies that contributed to the list. So there's definitely a challenge in terms of translating this data and validating our models is how we might best optimize our in vivo positive list to improve confidence. And again, I'll actually, there are a couple other references here, Ashner 2016 and Harrell 2018 that I would encourage you to visit to also learn a little bit more about the in vivo positives that we used.
Okay, great. I might have missed this. I'm sorry if you said this, but there's a question about including microglia in the test battery. Are they included? And if not, do you have an opinion about that?
No, they're not currently included. I did mention it as sort of, I guess I feel like maybe it's more of a pie in the sky dream at the moment, just because a lot of these current assays are fairly high throughput. So I have been really interested in hearing about the current state of available microglial assays. And I heard a little bit more about this at SOT, which was exciting. And so they're not included in this data set, but it would be something important to consider in the future and kind of put emphasis on getting an assay up to the point where it would be, on the throughput for chemical screening.
Okay. There's a question about whether the DNT NAM battery will be included in ToxCast, and I think it's about data access over the dashboard. Wondering if that's going to be something available in the future.
Yeah, so we're really excited. The summer release of the ToxCast data will include all the data that I showed you today. And if anyone is interested in using this data, it is publicly available in my manuscript. So you can pull the data from my supplementals there. But yeah, so the public release will include the network formation assay as well as the high content imaging assays that are starred here and will be publicly available.
Okay, and I'll just say, and this addresses one of the questions too, was I'll put that publication as well in on the webinar page so people can find it.
Perfect.
One last question. We have 30 seconds maybe. Wondering if you have any thoughts about using the battery. Is it sort of a positive screen or a negative screen in terms of using the results and then deciding on what to do with them?
So I guess if I understand the question, I would say the strength of the battery right now is is capturing highly sensitive effects. So, if anything, I think the biggest limitation is sort of interpreting the data across all of these assays, and how do we create some sort of threshold or high confidence rating? And do we say if there was activity in 1 out of 50 DNT NAM endpoints is that informative. So I think it's good at capturing, sensitive effects. And I think right now we're working on kind of capturing more biology as well as reducing the data set to the point where we can, efficiently use endpoints that we, know are informative. And there's a lot of sort of computational analysis that goes into it that needs a lot of tweaking. So yeah, again, so if I understand the question correctly, I think, it captures positives, and I think that it'll be certainly useful in using it for building predictive models in the future, and more chemicals and more biology are critical to improving the model.
Great. Okay, well, that will be the last word. Thanks again, Kelly, for your presentation. Thanks, Sabina, as well, for sharing your work with us. And thanks to all of you for joining and for your great questions. And we will see you next month. Bye-bye.
Thank you.